I'm going to introduce the third presenter under the theme of molecular and immunological diagnostic tests of COVID-19, current status and challenges. Associate Professor Hakho Lee of MGH Scholar Center for Systems Biology will deliver the presentation. He graduated from Seoul National University, majoring in physics, and also he earned a doctoral degree at Seoul National University at the same major, and now he's an associate professor at Harvard Medical School, and at the same time, he is a team manager of bioengineering management at Center for Systems of Biology. Good afternoon. I'm Hakho Lee, working at MGH in the United States. Our lab's major area of interest is technology development for easy and early detection of cancer and other infections. And regarding the COVID-19, we are also putting even more effort to develop a diagnosis technology. So far, we have had an opportunity to look at the current trends of diagnosis technology and found some room for improvement. And I'm going to talk about such trends and areas that need improvement.
먼저 본론에 들어가기
Before I start, let me briefly talk about the COVID-19 diagnosis. And these are some reference materials. As you can see here, diagnostic technologies or methods and FDA-approved technologies are available. So data on these are available on our website in the form of infographics. So details and technical parts of diagnosis technologies are published in iScience, which is a review paper. And regarding what diagnosis technology we should apply and the context, at Science Translation Medicine, we published a perspective article. So for further information, please refer to them. The trends of diagnostics technology or statistics are available in the websites below. To respond to the COVID-19 pandemic, diagnostic technologies are very much important. The importance of such technologies have been emphasized these days. So one of the unique characteristics of the COVID-19 is that asymptomatic patients or patients with mild symptoms could also transmit the virus to others. This is the biggest difference from the SARS outbreak we had before. So when it comes to SARS, if patients start having infectivity since when they have shown some symptoms, so that health authorities can isolate patients after they've shown symptoms so as to stop further spread. So as you can see, 140 days after the first SARS case was reported to WHO, the SARS curve had almost been flattened. And as far as I know, there has been no single SARS case so far. However, in contrast, asymptomatic COVID-19 patients or those with mild symptoms have already infectivity. That is why in just 20 days, the number of confirmed cases has dramatically surged after the first case was reported. Due to this virus's unique characteristics, early and accurate detection and diagnosis is very much important. And there are two ways to diagnose COVID-19. First, as you can see here, you can collect samples through nasopharynx, and you can detect the viral RNAs. The other way is that you can collect samples and detect antibodies to the virus, which is called immunodiagnosis. So depending on the virus context, depending on what you want to detect, test methods vary. Nuclear acid amplification is to detect viral RNAs for early detection or confirmation. And immunodiagnosis is to detect antibodies so as to check whether a patient is recovered from the infection or has immunity to the virus. So the method is also to check whether a patient is able to go back to the society or not. So depending on the context, tests should change. This is important. So through my presentation, I'm going to talk about these two tests. One is nuclear acid amplification, and the other one is immunodiagnosis test. There are two methods for nuclear acid amplification. First is RT-PCR test that regulates the temperature to amplify nuclear acid. And the other one is isothermal PCR that amplifies nuclear acid at the same temperature. As you are well aware of it, RT-PCR is the golden standard, which means it is the standard test. And as you can see here, a unique gene sequence will be used for RT-PCR tests. So this picture shows how a test proceeds. You put a swab into nasopharynx to collect samples. The use of swab is recommended, but it is not a cotton swab. It is called a flocked swab. There are strands at the tip for enhanced sample collection. So the use of flocked swab is recommended. After collection, the specimen should be transported so that it is put into a viral transport medium so as to be transported to a lab. It is recommended that the samples be delivered within 72 hours and stored at the temperature between 2 to 8 degrees Celsius. Once specimens are arrived at the lab, then RNA extraction proceeds, which takes about 1 to 2 hours. After the RNAs are extracted, RT-PCR proceeds, which takes also about another 1 or 2 hours. So after 3 or 4 hours after the samples are delivered to the lab, the test result will come out. So it is done in a lab, larger lab, not on site. So big laboratories can conduct RT-PCR tests. So to make this kind of RT-PCR available on site, so many technologies are being developed. And one of them is isothermal PCR. To conduct an isothermal PCR test, you put samples and reagents in one tube and maintain the same temperature. During that process, nuclear acid will be amplified, and as you can see, signals could be detected. So this method is a very good method to use on site. Indeed, with this kind of isothermal test, Abbott released a product called ID Now that applies isothermal amplification. You can see cartridges for ID Now here, and this is the device. So you can put samples using cartridge and put that cartridge into the device, and you can detect the virus within 13 minutes. So one of the technological benefits of isothermal test is compact instrument, and it can be a point-of-care method. And when it comes to sensitivity, it is similar to PCR. But if you use isothermal method in clinical tests, results would vary. As you can see, this slide shows the test result of ID Now applied for 101 patients in a hospital in New York. The results showed that there were many high false negative cases. So the level of sensitivity was just at 52%. It means that the isothermal method is relatively new one, so it needs to be further validated and verified. So when it comes to nuclear acid amplification, in summary, currently RT-PCR is the most important and principal method at this moment. The supply chain for RT-PCR, including reagents and equipment, is well established. And the process of primer design or probe design is relatively simple, and which could contribute to lower test costs. And also RT-PCR can conduct batch process. As many as 384 cases could be handled at a time. And most isothermal PCR related technologies and reagents are still protected under patent. So there are some sort of monopoly. In contrast, RT-PCR is royalty-free, which makes the method RT-PCR the most suitable one for test. So far, I have covered nuclear acid diagnosis, and from now on, I'd like to move on to immune diagnosis. What we want to detect from immune diagnosis is antibodies that are developed in the host after infection. We want to detect IgG or IgM. So these antibodies are developed in the body after viral infection. To detect such antibodies, when developing assays, we use antigens. We put artificially created antigens on a plate so as to detect antibodies. And then we may check the color or fluorescent colors to generate signals. So above all, the most important thing for antibody detection is to find and have effective antigens. During the SARS outbreak and current COVID-19 outbreak, a lot of things have been discovered from research studies. This is a picture of a virus. When a virus gets into the body, then immune reactions are induced by a protein called spike protein or S protein. And nuclear capsid or N protein also induce immune reaction in the body. Therefore, to detect antibodies generated by COVID-19, using S or N proteins is the most effective one. Then which one should we use between S and N protein? So regarding that, the US CDC carried out a study. Here is a table. So it shows the comparison between COVID-19, SARS, MERS, and the common cold virus. So N protein and S protein of those viruses are compared. So S protein is broken down to S1 and S2 unit, and the table shows how much similar viruses are to each other. So for example, COVID-19's S1 protein is 100, and the level of S1 protein of MERS is 24%. So as you may find out from this table, so antibodies created due to the COVID-19 could be captured the most when using S1 protein because S1 protein has the minimal cross-reactivity. The US CDC compared the ordinary people, healthy people, common cold patients and coronavirus cold patients, MERS, SARS patients, and COVID-19 patients. They compared blood samples of them using S1 protein as a capture antigen, and they compared signals of them. As you can see, 4 groups showed no signal at all, and SARS and COVID-19 patients showed some signals when using S1 protein. So the results suggests that to capture antibodies to COVID-19 using S1 protein is the best way. Of course, some of you may say SARS and COVID-19, these two have cross-reactivity to one another. Yes, you're right. Those viruses are similar to each other, therefore they have cross-reaction. But the further outbreak of SARS has never happened ever since the first outbreak occurred. So I believe that among the population, I believe almost no one has SARS antibodies at this moment. So therefore, there's no need to worry about cross-reaction between SARS and COVID-19. So to carry out this kind of antibody test on site quickly, many rapid diagnostic tests, so-called RDTs, using retroflow are being developed. So the test can be used to estimate how widespread the virus was and to decide whether individuals can return to social contacts. But there is one issue. A lot of RDTs are now being developed, but as you can see here, the performance deviation between them is quite large. So when it comes to sensitivity and specificity, deviation is quite large. So the imperative thing is to develop an effective sensitivity and specificity test. So far, I have touched on nucleic acid amplification diagnostics and immunodiagnosis. Then for the rest of the time, I'm going to briefly talk about how we can further develop these diagnosis methods. One of the lessons learned we have so far from the outbreak of COVID-19 is that the development of antigen test is very important. All tests that have been carried out so far have been done with PCR, so we need new test methods to reduce PCR test volumes. In fact, antigen tests for influenza have already been conducted. So there are test kits for quick assessment. So if the kit says it's positive, then treatment process can begin immediately, and we can recommend RT-PCR test only to those who have negative. So with this kind of technique, we could reduce workload for RT-PCR. To that end, we need new test kits for accurate and quick detection of COVID-19. At the same time, RT-PCR system is the basis of molecular diagnosis, and we can make it faster and quicker. And now new technologies to make RT-PCR quicker are being developed. As you can see here, one of them uses light. Like this, we need to make the existing RT-PCR faster. And as I mentioned before, when it comes to antibody tests, it's imperative to develop more effective and accurate antibody test methods. And through antibody tests, we should acquire accurate data on how prevalent the coronavirus is, because we can make a decision regarding how much more accurate tests should be. All of them are interconnected, so therefore developing accurate tests like serologic test is one of the many challenges. Then let me summarize today's presentation. So there are 3 key takeaways. To respond to a disease that has no vaccines like COVID-19, we need to carry out quick and extensive tests. This is a lesson learned. And depending on the clinical context, whether it be diagnosis or recover, we need to carefully choose right test depending on the clinical context. And when it comes to technology, still, and we need to develop one that could be mass produced and that could be used on site. And as I mentioned before, for further information, please refer to materials I showed you before. And plus, please visit our center's website and please refer to papers on the slide for your reference. And thank you very much for your attention. And I wish you stay safe. Thank you very much for your attention. After Professor Lee Hakho made his presentation, there was a question raised. Are there, if any rapid kit, if a rapid kit is developed then for the ordinary people, then would be able to ease the burden of medical staff members. So regarding that question, if a rapid kit is developed, then ordinary people, I believe ordinary people will be able to ease the burden of medical staff members today. But that kind of rapid kit, to make it available to ordinary people in the market, then molecular diagnosis, so sensitivity and specificity and accuracy must be equal to that of the existing methods. For example, let's take influenza as an example. We have a rapid kit for influenza. So far, we have developed many antibodies. So about 90% of molecular diagnosis sensitivity is secured. So far, many hospitals and other clinical centers are using that kit. So when it comes to COVID-19, so molecular diagnosis like technologies, if such technologies are developed, then we will be able to ease the burden of medical staff members for sure. So we are so looking forward to that kind of kit. So this is the answer to your question. So if needed, I will ask questions to Mr. Hakho Lee. Thank you for your question.